Hello, everyone, and thank you for being here today and listening to this presentation. I'd like to first thank Jack Parallel, my fellow researchers, Jack Parallel, Gabe Del Monte, and Dr. Ann Kirkton and Janine O'Brien for helping this molecular biology course be possible. So I'll start by giving a little introduction to what we were researching. Um, Snowflakes are very beautiful and something that has long interested scientists and researchers around the world. So I'll show you a little bit about how bacteria um, can form a nucleus of the snowflake and how oftentimes snowflakes need a small particle such as dust or bacteria, which you can see in figure 1 here. So that bacteria will form that central little point that water molecules can essentially grow around or form this ice crystal around, which can be seen in the second part. And after this ice crystal gets big enough, you have a snowflake that is falling to Earth or down to our surface, and that it can contain dust particles or small particles as well as bacteria. So this can have implications on our health, on a variety of different things. So it's a very interesting research topic that we're talking about. So the next thing that I'm going to talk about is a little bit about Duluth's environment. So we have a lot of lake effect going on. And what this means is that cold, dry air, generally from the north, comes across the lake and it picks up a bunch of moisture. This moisture in the wintertime, it's converted into snow, and it is often deposited on the shore of Superior, right here, and Duluth. So we have a lot of different snow that comes in. So we have plenty of materials for our research. So now I'll talk a little bit about the research question that we have. So we have, do snowstorms from different geographic origins carry different bacteria nuclei in their snowflakes? And does the snow that was collected contain bacteria in their snowflake nuclei? And do the snowflakes contain ice nucleation proteins? And the hypothesis that I have is if the snow that we collected contained different snowstorms origins contain bacteria, then the snowflakes will contain bacteria that also originate from different geographic locations, which should make sense if snowstorms are forming in one area and the bacteria are used for those snowflakes. Then it should make sense that those bacteria can come into a different origin or a different environment that they're deposited in. So this picture right here is a bacteria nuclei at the center. So this is just a snowflake, and we can't tell for certain, but this is the nuclei right here, and that could either be a snowflake with a bacterial nuclei or a dust particle nuclei, and that's just a great picture that shows that under high magnification. So next, we'll talk a little bit about the methods. So we first obtained snow from our environment. We then filtered this snow through an AeroPress with filter paper at the bottom of it that allowed our bacteria to stick on this filter. The filter paper was then put in tube 1 here, it shows, and we were able to use DNA isolation techniques to isolate our DNA from the snow. And we used a stool kit because the environment has a variety of different bacteria, and our digestive system also is able to harbor a lot of different bacteria that are very hardy. So a stool kit was a very good kit for this DNA isolation. We also did 16S PCR as well as gel electrophoresis for both 16S and ice nucleation proteins. Next, I will talk a little bit about the methods section here, or the results, my bad. So to begin, on the left here, we have 16S, and this is a gene right here that's present in one of our 23BT samples. And this is really cool. So it shows that bacteria is present in this sample. And this is our positive control. So it shows that 23BT snowflakes contained bacteria in them, which is pretty cool. The other two didn't, or maybe not enough to be shown. The one image on the right here shows our gel electrophoresis results after PCR. this shows our results for 22 BP. This is a sample that had most likely had an isonucleation protein. So this band is present, and that means that this bacteria most likely has an isonucleation protein, which is very applicable for our environment for creating snow at lower temperatures, as well as ski resorts that might want to use this bacteria to help them earn a little bit more money and basically help them create snow at lower temperatures than normal. So I'll get rid of my annotations quick. Okay. So the next thing I'll talk a little bit about is future directions. So I talked a lot about how we were able to address our questions through our PCR, our gel electrophoresis, DNA isolation. But the next steps would be metagenomic sequencing. And we would use Illumina sequencing most likely. And you can read a little bit more in that second paragraph of this section to learn a little bit more about Illumina sequencing. But we would sequence the entire genome of our species to be able to tell, okay, what species is coming from each location. We would try to, we have the directions and the origins of the snowstorms that we collected our snow from. So we know that, okay, if this snowstorm came from the north, now our next thing is say, what type of species often are carried in the snowstorms in the north? Or what type of bacteria would be contained in the snowstorms coming from the south? So this would take Illumina sequencing to sequence the entire genome of the bacteria so we can know that and address the second part of our hypothesis, because we were able to address the first part that bacteria are contained in the snowstorms and that they do contain ice nucleation proteins. But we do need to do further testing to fully confirm our hypothesis. But to conclude, based on our results, a little bit more testing is need to be done to confirm our hypothesis fully, but we were able to accomplish a lot of things in this molecular biology course through our techniques of DNA isolation, PCR, gel electrophoresis, and everything that we did in this lab helped us get a very good understanding about the many techniques that you can use. And I'm really excited to see the future results, hopefully, of Illumina sequencing and seeing what type of bacteria species are contained in our environment. Thank you very much for listening to this presentation.